So in this video, we'll be looking at how fatty acids are utilized within cells to produce energy. So the first stage of this process is the transport of fatty acids from the cytosol of the cell into the mitochondria, where the beta oxidation machinery is actually found. And now this transport process is not a simple transport process because the inner membrane of the mitochondria in particular is quite impervious or impermeable to the fatty acids. So we need a transporter or a translocator in this case to kind of help shuttle our fatty acids into the mitochondria. And the way in which this particular system works is by using carnitine. And carnitine is quite a useful molecule, as you can see from this diagram, because what we can do is we can activate the free fatty acids by coagulating them. We can add an acetyl coenzyme A group on here onto the fatty acid, and this is done by a sulfhydryl linkage, and that activates the fatty acid. Then we can use the carnitine acyl transferase system, where we can replace the coenzyme A with carnitine group, and then the carnitine acyl molecule can be transported via the carnitine translocase into the mitochondrial matrix itself, where we can remove the carnitine and put back on the coenzyme A group, which leaves us with our transferred fatty acid, this time inside the mitochondrial matrix. And now this is quite a useful recycling system of carnitine itself. So the transporter on the inner membrane of the mitochondria is a translocase. So the acyl carnitine comes in and the free carnitine goes out over the membrane as well. And by this process, we can recycle the carnitine molecule as part of the transporter process. So in summary, the activated coagulated fatty acids have a coenzyme A replaced by carnitine. This is translocated over, carnitine is removed and shuttled back across as part of the transport process. Now, our fatty acids are formed as containing coenzyme A, so we have to first activate them in this process. So what we have here is a free fatty acid represented by this kind of pinkish gray you can see here, and we have the R group, which I'm sure you'll be aware is just a hydrocarbon chain, and we have the carboxyl acid group represented by these oxygens here. And what you can see is that when we introduce ATP, we form, thanks to our fatty acyl-CoA synthetase, we can form an intermediate that's bound to the enzyme that's adenylated. So it has adenosine phosphate attached to the carboxylic acid group, and this can be replaced by coenzyme A, which leaves us with our fatty acyl-CoA. Now, this process does require a little bit of thermodynamic driving, and so what we also couple it with in order to achieve that is the lysis of pyrophosphate into two inorganic phosphates. So in summary, ATP is used to adenylate to produce an intermediate, which with the help of pyrophosphate being degraded into two inorganic phosphates can then be replaced by coenzyme A group, giving us our activated fatty acid, which can then take part in the carnitine transport pathway we discussed in the previous slide. So this allows us to get our fatty acid in an activated form into the mitochondria, and this is where the beta oxidation can occur, because this is where the beta oxidation machinery is found in the mitochondria itself. So this is an oxidative process, hence the name beta oxidation, where we take the fatty acyl-CoA, we oxidize it, hydrate it, oxidize it further, and then remove the thiol groups and remove the coenzyme A. And this leaves us with an acetyl coenzyme A and a shorter fatty acid CoA molecule as well, which we can feed back in at the top here and go round and round the cycle again and again. And this is really what underpins the energy density of fats. These fatty acids, they can be 18 plus carbon chains long, which means that there's a lot of 2 carbon molecules that can be chopped off in the form of acetyl coenzyme A, and we'll see in the next video how much ATP that can actually yield. And in the next video, we'll kind of go into the individual reactions of beta oxidation.